in the second part of this presentation, we are going to focus on how do you detect and how do you characterize an outbreak? How do you detect and how do you characterize an outbreak? Now, to detect an outbreak, you need case definitions. Who is a case? Who is a case? Who has this disease that you're talking about? So you need a case definition, both for new diseases, even new diseases that you've never seen. When Ebola had just come, nobody had ever seen it. But they had to come out, when they saw the first cases, they had to write some rough case definition. Now, also for existing outbreak-prone diseases, you need to have these case definitions and disseminate them to as many people as possible so that anybody who suspects a case can quickly raise an alarm bell. So that's number one. Number two, you must demonstrate an unexpected rise in new cases. That is the rise which exceeds a certain level. And how do you know that it exceeds a certain level? You need a threshold. You need to put a bar, a ruler, and you say, beyond these cases, we are having an outbreak. Now, these thresholds are not the same for all outbreak-prone diseases. They differ. Now, for cholera, for you to say there's an outbreak of cholera, you need one confirmed, focus on the word in blue. It has to be confirmed. So when you have one confirmed case, so you have the case definition for cholera, you have profuse watery diarrhea, a lot of a person coming with the profuse watery diarrhea, extreme weakness, sometimes with vomiting, and they get rapidly dehydrated in a short period of time. So that is a clinical definition. Then you take off their sample, their stool sample, you take it to the reference laboratory, they check and they confirm that this is the bacteria Vibrio cholerae. Once they confirm one case, that's an outbreak. You don't need to get 5 cases. If you wait for 5 cases, everybody will get cholera. So that's one case. Now, for Ebola, and that's the interesting one, one suspected case is an outbreak. You have to run away fast and you don't come and say, okay, let's take off the sample and go to confirm. No, you have to first retreat, do the necessary protection, put on the protective wear, send a trained team to come and take the sample, then go and test. So one suspected case is an outbreak. So it's not uncommon to hear the Ministry of Health announcing, like recently, that there's an outbreak of Marburg, suspected Marburg in Queen District. So they don't first wait to confirm. Once it is suspected, it fits the clinical picture, then there's a probable outbreak. Now for measles, you need 3 cases from one parish. One parish is an administrative unit below a sub-county in a week. If you have 3 cases from a parish in a week, or if you have 25 cases occurring in a health sub-district. A health sub-district is a health zone of about 100,000 people. Then you say measles. Now, meningococcal meningitis, if you get 10 cases in a higher risk district, those districts in the meningitis belt, in Uganda, it's Yumbe, Adjumani, Arua, Terego Maracha, and so those districts in West Nile, probably the northern parts of Nebi district and Zombo district. So those in the meningitis belt, you need 10 cases. While for less risk areas like central Uganda here, you need 5 cases. So you need fewer cases. Now for STDs, sexually transmitted diseases, these are constantly with us. So you just need a slow or sudden rise in prevalence beyond the seasonal trends for STDs. And the same for malaria, when you get a sudden or slow rise in prevalence beyond what you expect for a particular season. Like we expect malaria to increase in the rainy season. So if it exceeds way much more, then we may talk of a malaria outbreak. Now, so just to recap, that for certain diseases, they occur constantly, they are endemic in the communities. For an outbreak to occur, you need a sharp rise in cases. An example is malaria. Then for some epidemic-prone diseases, they don't exist normally in the community. So occurrence of just one confirmed case is considered an outbreak. Example, cholera. Then for other diseases that are rare and highly deadly when they occur, just one suspected case is enough to consider an outbreak. Now, how do you recognize an outbreak? How do you recognize an outbreak? Now, epidemiology, if you remember from the definition, it is the distribution and determinants of health-related states in populations. Epidemiology is an observational science. You must be able to observe. You will not move around with a microscope everywhere. You have to observe for abnormal event. So that's why we say astute observation. Astute meaning you are attentive. You question things. That's when you see an outbreak. If you are not astute, by the time you detect an outbreak, it will have killed several people or affected several people. So you observe, sometimes a single event will trigger you. Like for Marburg, Ebola, you get a case that is bleeding from the nose, that is, dramatically deteriorating. Don't wait, a single case. But in many cases, we observe a cluster of events because we need to be tickled several times to say that there's a problem. Now, for non-epidemic prone diseases that have been with us, like cholera, watery diarrhea, dysentery, the immunizable diseases like measles, the signs and symptoms of these from the case definition occurring in a cluster of cases is what should guide you. If you see many children coming with a papular rash in their foreheads, with red eyes, with a cough, coryza, and conjunctivitis, that is the red eyes, then don't wait. It might be measles. So a cluster of such cases. Now, the problem is with a strange disease because you may not have seen the symptoms. So for a strange disease, you observe strange symptoms that are not exactly matching with what has been occurring. They could have occurred before, like bleeding from the nose, it happens. But the way they are combining with other risk factors does not add up. Then you start suspecting. Now, these observations come from clinicians or laboratory personnel. Those are, in good surveillance system, we would think that clinicians and laboratory people would be the first. But unfortunately, in our health systems that are a bit weak, we rely on other people, like community leaders and opinion leaders. They announce. Rumors. In epidemiology, epidemiology is one of the few disciplines where we love rumor mongering because it is through rumors that we get to know many of these epidemiological events that would kill so many people, that we know them in time. So rumors from the community. Now, sometimes these people in the community do not call the district health officer. They call directly the Minister for Health or their MP and they say, oh, there is this strange disease. So all those sources are important. And then patients, one or more patients may report that there's a problem. But then also the routine surveillance activities in the health facilities. And that is the one that is desirable. If we can strengthen our surveillance activities so that they are as strong as the kind of surveillance that is done in security and the military. Now, one illustration, strange disease. Strange diseases are a little bit more problematic. This was in DRC in 1976 when the first cases of Ebola first emerged. This is what happened in Yambuku Mission Hospital. So it's not by surprise that the first place here was Yambuku Mission Hospital. 17 patients and a midwife had died since September 1st. This was September 19th. So roughly every day they were losing a patient, which even in very busy hospitals, if they are district hospitals, it's very hard to get a person dying every day. But here, 17 people died over a period of about 19 days, and they all had similar symptoms: fever, vomiting, abdominal pain. These are fairly common symptoms in outbreaks. But in addition to that, they had bloody diarrhea And they were, so bloody diarrhea, you may think of other outbreaks that cause bloody diarrhea, but in addition, these people were hemorrhaging. They were passing blood from all other orifices, from the nose, from the eyes. So, and the illness appeared to be spreading to the hospital staff, because usually these strange diseases, they start from community members, but they will spread to the hospital staff. So, the incubation period of a disease is also quite important. We are interested in knowing for a disease, when did the exposure begin? When did this person get contact? So, like for Ebola, if we get a case, we ask, who are all your contacts? Who are the people you got into contact with? And if one of them also suffered from similar symptoms, we say, when did you first get in contact with that person? And then, when did the disease begin? And therefore, what is the incubation period? Now, the incubation period is the time interval between when a person is invaded by an infectious agent to the time when they get symptoms. Now, the incubation period is quite important because very often, during that time, infection can occur. So we can get people being infected, but there are no symptoms. So a person has no symptoms, but is infectious. So it's quite important. But also it is important later, we shall talk about characterization of outbreaks, and we see the importance of the other importance of the incubation period. Now, characterizing outbreaks. We characterize outbreaks using what we call epidemic curves. They are a plot of cases over time. Like, for instance, if my time is in days. So on Monday, how many cases did I get? On Tuesday, how many cases? On Wednesday, so it's a frequency plot. And from that, we draw a curve. Now, there are two types of epidemic curves that characterize outbreaks. There's what we call a single exposure common vehicle epidemic curve and a multiple exposure curve. And we shall look briefly at these. In a single exposure common vehicle epidemic curve, also known as the point source epidemic, we tend to have a curve. So you start with a case, a few cases, and then there's a sharp rise in cases up to a peak. Then it goes down. And usually the peak time, the time when cases are maximum, corresponds to the median incubation period. The median incubation period. So that means that many cases occur during the incubation period. People are being infected during the incubation period, and then at the median of the incubation period, you get the maximum number of cases, and then it goes down. Now, what does a point source epidemic mean? When you get such a curve, when you get such a curve, it tells the epidemiologist that there is a point source where these infections are coming from. If you get a curve like this, it means there is a point source where people are getting the infection from. So your role is to look for that point source. That point source could be something that is contaminating the water that all these people are drinking. It could be one person that is infecting everybody. Let's say a man who is moving around and sleeping with as many people as possible with unprotected sex, and maybe they have gonorrhea and they are causing an outbreak. So you have to look for that man, the index case. It could be anything. So the key thing here is that there is a point source. So the importance of when you get this curve to the epidemiologist is that you are supposed to look for that point source, and when you deal with it, the epidemic will be controlled. This is an example of a point source epidemic curve that I was involved in an outbreak that I was involved in investigating in 2004 in Kayunga district, which is in central Uganda. And initially, based on the symptoms that the people were getting, we thought it was cholera. They had diarrhea, they had a lot of vomiting, and they deteriorated very fast. And in a short period, they were dehydrated, and some of them died. So we went in to investigate, and we went through the outbreak investigation process. And what did we find? we found that there's a certain tribal group there that has certain behaviors in dealing with the dead. So when after burial, they undertake cultural functions for several days, sometimes stretching in 2 weeks. And they carry some food, especially starchy foods, that will stay for a long period of time. And we found that almost all the people who were affected in this outbreak and those who died had contact to a certain meal of cooked sweet potatoes that had been stored for several days. And what did we have to do? We looked for that particular household that had that meal, confiscated the meal, took some samples to the National Chemotherapeutics Lab to test, and indeed they found that the meal was infested with Clostridium botulinum, which is the cause of food poisoning. And then after that, we confiscated several other meals and then health educated the people on the dangers of keeping and storing such food for long periods of time. So once the point source was dealt with, the outbreak immediately went down, and you can see here that it went down very, very steeply. So that is an example. And another curious thing to note in point source epidemics is that you usually have the first case. This one is called the index case, the first case. And that case, in the case of Kayunga, died. So the cases went to 0. And then there was a period of time when there were no cases, maybe from 1 to 1.5 days. And then all of a sudden, there was this bang, and cases started to increase. So that's how the point source epidemic behaves. Now, the other type of epidemic curve is the multiple exposure epidemic, or the propagated epidemic. Here, in such epidemics, we don't have a single point source, but we have several point sources. So there are several point sources, so the outbreak keeps getting propagated, you know? So it's in such outbreaks, it's difficult to control, because there may be, not only are there several point sources, but the point sources might be different. One could be a human being. Another one could be a contaminating site. Another one could be a water body. So there could be several point source epidemics. In Uganda, we suffered from several propagated epidemics of cholera. One good example is in the Lake Albert region, in the escarpments there. And when you visit that place, you can really see why these epidemics are propagated. The latrines, they tell people to construct latrines, but the latrines are very difficult to construct. You go only 6-7 feet and you find water, so they have to construct the latrines upwards, and it is costly. You have to get bricks, so they are relatively shallow and they fill very fast. And then some of the sewage is channeled through small channels, and it ends up into the lake. In the lake, there are people who are washing, there are people who are drinking water, there are other people who are defecating or urinating on one side, and it is all in one ecosystem. Now, when the healthcare workers go there, they tell these people to boil the drinking water. But when you look at this place, for almost a 5-kilometer radius, firewood is quite rare, because down there in the escarpments, many of the trees are shrubs, and they've all been destroyed because of the big business of fish and the drying of fish. So there are no firewood. There's no firewood. So how do you tell people in such an area to boil their drinking water? So these are some of the challenges that could propagate these epidemics, and they are quite difficult to heal. This is an example of the cholera outbreak in Maputo between 2000 and 2001. Now, there are some key terms here that you need to know about epidemics. About characterizing epidemics. There's what we call a common vehicle exposure. It means that there is a point source, and all cases share that point source, whether it is one spouse or one sweet potato meal or one vector. A vector is moving from one person to another and infecting, so they share it. And a single exposure means that there is one, not only is there a point source, but a person is exposed to it once. They eat the sweet potatoes once, and bang, they get sick. But multiple exposures means that there could be one exposure, one source, but people get exposed to it several times. They eat several meals. So they increase the dose. They increase the dose, and that's when you get the outbreak. And then there's what they call periodic exposure. Periodic exposure means there's a contaminating source that releases the contaminant periodically, intermittently. Like, for instance, a sewer pipe which bursts during the rainy season and it keeps releasing. But continuous exposure means there's constant contamination. Like, for instance, many of the surface wells here in Kampala are contaminated with the fecal coliform bacteria. Now, this leads to the two types of epidemics that I talked about, the single exposure, common vehicle outbreaks, and then the multiple exposure epidemics, which lead to the two epidemic curves. Now, the last thing I want to talk about in this subsession is the issue of measuring outbreaks. Now, as you know, to measure the ferocity of, outbreaks, we use incidence. Incidence, if you remember, is the number of new cases occurring in a particular population over a given period of time. So incidence is new cases over the population at risk over a period of time. And usually, sometimes we compute incidence for 1 year. And then we say the annual incidence of this particular disease is this. Now, in outbreaks, so this is incidence. In outbreaks, we use the same parameter, but we ignore, the period of time is implied. Because an outbreak occurs within a short period of time, we may not wait for a year. So we consider that period of time when the outbreak is occurring. So the time is implicit, and we only consider the new cases over the total population at risk in that area. And this special form of incidence, where time is implicit, is known as the attack rate. And that's just that. So that's one of the key measures. But there are other important measures like the case fatality rate, the proportion of those who acquire the disease that die. So the case fatality rate is another important measure, and it could tell you how virulent the disease is and how deadly the disease is. But it could also tell you how effective the health system is in treating those who are sick. So apart from the attack rate, there are other parameters like the case fatality rate. So this is the end of the second subsession of this presentation.